So you all should see you.
You can see us.
Okay.
Wave to the internet, everyone. Hello, internet.
All right, and I need to pull this up.
There we go.
And just to make sure that you can see what I see, which is my presentation coming up.
If you could maximize that somehow, that would be great. Is that as big as it can get on your screen?
Yes. Is
Okay.
it? So what does it look like to you? Does it look like a full screen thing, or does it look like?
It's sort of small.
Oh, I see. So it's not pulling up the whole. All right. Is that better?
That's a bit better.
Okay. I think that's as good as we're going to get. I tried to put it in the full screen mode, and it's not. I don't think it's sending it to y'all.
That's the best as it's going to get right there. So let's just run with that.
All right. That sounds fine.
Okay. Let me give an introduction, and then I will let you take it away.
Okay.
All right. Our speaker this evening is Dr. Lindsay Waldrop. She got her undergraduate degree in biology with minors in physics and chemistry from the University of North Carolina at Chapel Hill. I believe she also had some mathematics background there as well. She got her PhD in integrative biology at the University of California, Berkeley, and her dissertation is interesting. It's the fluid dynamics of odor capture by crabs, hence the YouTube video earlier. And she's currently a postdoctoral fellow in the mathematics department at the University of North Carolina at Chapel Hill. And she has her presentation this evening called Tiny Heart, Big Controversy. What is the pumping mechanism of the invertebrate embryonic heart? And again, we want to thank the University of Central Arkansas's Foundation for the financial support for this seminar series. So take it away, Dr. Waldrop.
All right, thanks. Well, it's great to be talking to you all and everybody along on the internet, whoever's out there. So I wanted to start off by, let me see if I can do this. There we go, acknowledgments. So my current boss is Laura Miller, and she is a joint professor in the mathematics and biology department, and this is really her brainchild more than anything. What I'm going to talk about, it's 8 years in the making. It builds on the work of a lot of really fantastic people. Austin Baird, who was a graduate student with Laura Miller, who is now a postdoc in math at Duke. Nick Batista, who is still a grad student here. We're working on some other really fantastic stuff. And Tiffany King, who is an undergrad here at UNC and is headed off to med school next year. So I will just say that I've been along for the ride sort of for the past 3 years, and this is sort of a culmination of a lot of people's work. So to get started, I do a thing called comparative biomechanics, and I'm primarily interested in studying how animals and plants interact with their physical environments. Specifically, I'm really interested in organisms interacting with their fluid environments, internally or externally. So for my PhD, I did how crabs sniff, which is an external fluid interaction, and this for my postdoc, I'm doing these tunicate and embryonic tubular hearts, which is an internal flow. And really what it boils down to is an interaction between a fluid and a flexible boundary. But I wanted to really start off telling you an interesting story. When I tell people I study the physics of organisms, they often ask me strange questions. And this was one strange question that I got one day, and it is a story about jury duty. So I was picked to be on a jury, sort of distressingly close to my qualifying exams in grad school. And I was picked from a pool of potential jurors to serve on a case. And during jury selection, what they do is you write out a little card about yourself, what your occupation is, your age, and everything. And then the judge asks each juror questions to make sure they can be impartial and serve in an unbiased way. And so he got to me, the judge, and he says, it says here you study biomechanics. He looks at me and he says, do you study how things fly? And I say, well, I know people who do. And he says, I heard that bumblebees can't fly. Is that true? And so it was an uncomfortably serious tone, too. So I wasn't really sure. It caught me off guard. And I said, well, they do fly. And he was like, well, I know that. I mean, I've seen them flying around. I just heard that some scientists say that they can't fly. And I just wondered what that was about. And so I had heard some about this, and I said, well, you know, I think someone said that they thinking like they flew like airplanes, and that they couldn't generate enough lift with their stubby little wings, so they must not fly. But you know, they don't actually. So they don't actually fly like airplanes is the problem, right? They fly, they have these wings that they move around, and airplanes' wings, we hope that they don't move around while they fly. It looks really different. And so they're doing a whole bunch of different things than what people assumed that they were doing when they made that up. And he seemed okay with that. When I got home, I had never heard about this. I went, and it's totally a thing. Look, Snopes even has an entry on Bumblebees Can't Fly, which is fantastic. But I think what it really want to point out about this is that this isn't an airplane. You know, it's a living organism. It's in some sort of environmental flow, and it's doing a lot of different things. And so when you approach a problem like that with the understanding of the assumptions that it's flying like a thing that we've built, oftentimes you get it wrong. And so another good example of this, and I just want to point out because it's really relevant, is the issue of these boxfish. So the boxfish is this little guy right here. He's got a weird sort of carapace shape. And years ago, Daimler Chrysler Mercedes-Benz made this concept car based on the low drag shape of this boxfish. Well, it turns out that, you know, they said some things about it. It wasn't quite true. They didn't really put it in the environment that it belonged in. And it turns out these guys aren't super stable like the Mercedes-Benz people hoped and believed that they were. They're actually really unstable, and that's good because what they do is navigate through a very complex reef system in which they have to make turns and dive away from predators and things like this, and they actually get worse drag than what you would think of a typically low drag fish like a shark or a tuna or something like that. So if you're interested in this sort of story, this article in Slate, A Real Drag, is all about the concept car and how it misunderstands the boxfish and why is it so important when you're looking at biomimetic stuff to have a biologist on your team. So the reason that I go through all of this stuff is that I want to talk about another sort of genuine scientific controversy in which I think that it's going to boil down to another misunderstanding of putting an animal, making assumptions about an animal working a little bit too much like a physical thing, trying to turn a bumblebee into an airplane sort of thing. So the motivation for this talk, and again this is about heart tubes, is determining this pumping mechanism of the vertebrate embryonic heart. If you don't know what a vertebrate embryonic heart is, just hang on, we'll talk about that next. But this is really going to inform the evolution of the vertebrate heart development and the fluid mechanics and how all these things play together to not only develop through the lifetime of an individual, but also how a high-pressure, chambered heart circulatory system could have evolved. And so what we'll do is review the two mechanisms by which this pumping might occur, which is peristalsis or dynamic suction pumping. We'll examine the assumptions and the definition of peristalsis that might be important, and then we'll review and model the physical conditions of this embryonic heart that are so important to getting the answer to this question right. All right, so first of all, I just want to review a little bit of biology. I promise I'll be quick. And this is just how vertebrate hearts formed. Very basic. So we're going to talk about two types of vertebrates. One are mammals, and I have a picture of a chambered heart here. You know, 4 chambers, pumps blood through the lungs, pumps blood through the rest of the very resistive circulatory system. And you're pretty familiar with this. The other thing I want to talk about today is zebrafish. And zebrafish, it turns out, have a heart that is pretty similar to ours, sort of half. So they have this heart that is both, and I'll pull this video up so you can see it. So it has a atrium right here that pumps blood into the ventricle that's right here, and that's it. So they don't have a 4-chambered heart, they have a 2-chambered heart. And they end up being a really important model organism because their hearts develop very similarly to ours. And also, you know, it's frowned upon to use a lot of mammalian, especially human embryos for these things. That's not allowed. So it's much easier to use a zebrafish, which is a bit simpler and easier to keep in the lab. So that is that. So that's what that heart looks like. So how did they form? They start as a tube. And it turns out that mammals hearts also start as a tube. And this tube contracts. It's a muscular tube that has a lumen that's basically the inside of what will be your four-chambered heart. And it drives blood flow. It's the first organ in the body to function, and importantly, every other organ is totally reliant on the blood flow that this organ will produce, the heart will produce, to develop normally. So it's a very important thing during embryonic development. So what happens to get it to the place that we know, the endpoint, the adult heart, is that this thing starts to loop. It starts to bulge out into what eventually will be the atrium and the ventricle, and it starts to loop so that this will eventually contract sort of inward, this will bulge out and then contract inward, and then you get to this adult heart. Well, mammals do about the same thing, except it's a little bit more complicated. They also do a mid-looping sort of thing, and this is sort of color-coded what's going to turn into what here. But this will twist around in an additional step and sort of lay on top of these other two things, and then a division will sort of grow in between that to form the four chambers. So it's got a couple of additional steps to zebrafish, but it's basically the same process, especially early on. And what we're going to focus on today is this stage right here, this tubular heart stage, and we're going to ask, how is this tube, this muscular tube, driving fluid flow that is so important for the development of the rest of the organism? So first of all, we have to sort of talk about and get on the same base about what peristalsis is. So if I could see you, I would ask who knows what peristalsis is or who thinks they can explain it. But since I can't see you, I'll just go ahead and go through the basics. So I have a tube here that could be a roller, a plastic tube, or a muscular tube, or anything sort of tube, because you can do it a bunch of ways. First of all, peristaltic pumps are positive displacement pumps. So they create fluid flow by displacing a fixed volume of fluid down the tube. Second really important point is that they have a non-stationary compression wave. So this is a wave of, if you squeeze it, if you would squeeze your hand, I'm trying to think of like something you would squeeze down a hose and run your hand down the hose to sort of evacuate the contents of that tube. That's kind of what peristalsis is doing. And three, there's no structurally fixed direction of flow. It only depends on the direction of this traveling wave. And so if you imagine blood cells in this tube, you can pinch the tube here and run it, and that will drive the flow in this direction. But equally, there's no reason that you can't do the same thing in reverse. There's no valves or anything like that at this stage of heart development specifically. So for muscular tubes, often they have circumferential muscles, which are these thread lines, and when these muscles contract, they decrease the diameter of this tube, and that's how, like your gut works, is a good example of peristalsis. It generally goes in one direction, but very unfortunately sometimes can go backwards. So there's nothing that prevents it from going backwards. And sometimes that unfortunately happens, very unpleasant. But that is basically the basis of peristalsis. Now, engineers have been using peristalsis to drive fluid flow for a really long time, and they use something called a roller pump. And so here's a little video of a roller pump. I'm going to loop that so you can see it. And as you can see, these little rollers go back and forth, and they create this sort of fixed volume that they're pushing through this tube, which is all kind of dull and boring. But when you study a pump like this, a mechanical roller pump, you come to a couple more points specifically about the fluid flow that this thing produces that have also been incorporated into a definition of peristalsis. And these points are that these types of pumps can produce continuous flow. So there's no pulsatile nature, there's no like faster, slower, faster, slower. It basically produces continuous flow. The flow speed that comes out of these pumps is equivalent to the speed of the compression wave. So the speed of this roller going around is basically the speed of the fluid that you get coming out of it. It also has the caveat that the peak flow velocity coming out of this tube or in the lumen of this tube does not exceed the compression wave speed. And the sixth thing is that there's a linear relationship between flow speed and compression frequency. So if you can imagine how fast this roller goes around, if you wanted a higher frequency of this roller, what would you do? You would basically increase how fast this thing was turning, right? So you would increase the wave speed, and that would correspond proportionally to an increase in how fast this fluid moves through the tube, right? So, but it's a little bit weird because biological pumps aren't always roller pumps. Functionally for this talk, we're going to break these points of the definition into two sort of sub-definitions. We're going to leave the first 3 points as the biologist definition. So it's just looking at the waveform going down the tube. It's not worried about what kind of flow is being produced by this waveform, just worried about the waveform. And then this whole thing, all 6 points, we'll call the engineer's definition because it's really based on these mechanical roller pumps that produce these extra flow features that are very consistent with roller pumps. All right, so now that we've got peristalsis down, we need to talk about embryo hearts. Okay, embryonic hearts. So this was a bombshell that was dropped. For over 100 years, biologists have been describing pumping mechanism of embryonic vertebrate hearts. You can see it here outlined as peristalsis. In 2006, a group in Caltech made observations of an in vivo, so a live, unmolested embryonic heart functioning as it should in the organism. They made observations that suggest it's not peristalsis, but this thing called dynamic suction pumping. And so the next thing we have to do is understand what dynamic suction pumping is to understand like how big of a shock this was to the biologists who studied this problem. So dynamic suction pumping. The basic setup for dynamic suction pump is this. It has rigid inflow and outflow tubes, so like a circulatory system, rigid inflow and outflow tube, and then this elastic, sort of really flexible section in the middle. Dynamic suction pumps work, they're also called impedance pumps. They work because the tube ends have a mismatch in impedance. So this is the resistance to the wave to propagate. And so you can imagine if you were here, if you sent a wave this way, it would resist that waveform a lot because it was close to the rigid edge, as opposed to this where it could travel sort of freely through the elastic section. And that is basically what happens. There's a single stationary compression site. So this is really different than peristalsis, right, where there's that traveling wave of compression sites. Single stationary compression site. And the compression site is also always at an asymmetric position along the tube. And the site position is actually what controls the direction of flow. So if you can imagine contracting your tube here, what happens is you'd get a bidirectional wave, and the impedance of that wave meant that it would freely propagate in this direction, but then reflect on these rigid inflow of the tubes. And the reflected wave is what actually drives most of your fluid flow there. And then it's reflected at the outflow current. So I have a little video of a dynamic suction pump that I made. This is a stiff glass raceway and a little piece of PVC tubing, and it's got some glitter and water in there. And this is my electric toothbrush, so I brought it in from home to make this video. And you can see that I'm pressing it sort of closer to this end, and it's driving quite a good bit of fluid flow through there, so I'll play it again. It's a really weird thing to think about, but actuating it at this single point will drive fluid flow through this tube, which is kind of cool. All right, so that's what a roller pump is, or I'm sorry, Liebau pump is. So here are the observations that this made, and I'm going to pull this movie up. Sorry, all this pulling movie stuff up. I usually do this in Keynote, but it wouldn't work with Google Hangout. So here's the video, and what we're looking for is that contraction at a single point, the bidirectional wave here, the reflection, and a suction bolus that's created by that reflecting wave, okay, that ends up doing most of the movement there. So here's a faster sequence of the heart pumping, all right, and sucking in that fluid. And it's got all these little markers for where they think the reflected waves are and what's happening. All right, so these observations basically show, all right, consistent with dynamic suction pumping. There's a single point of contraction, there are bidirectional waves, There's a hydroelastic suction mechanism. All right, and this is sort of outlined here at the bottom. So this is from their paper. They also, at the same time, saying that they had all these observations that were consistent with dynamic suction pumping, they also said, okay, we also have all of these observations that are really inconsistent with peristalsis. They're violations of peristalsis in the measured blood flow that we had. And I have this graph of average blood velocity here that's measured through this heart. And then the heart rate that they varied by putting the embryos in different temperatures. So if you increase the temperature in the area of an embryo, the heart rate will increase. They said that they could find pulsatile blood flow, that the peak blood flow velocity exceeded the speed of the contraction wave. So this dotted line is the speed of the contraction wave, and you can see that there are velocities that clearly violate that rule. And that there's a nonlinear relationship between heartbeat frequency and blood flow velocity. So these are the three parts of that engineer's definition of peristalsis that they say these really violate. Here's that nonlinear relationship. Really crazy, right? This is definitely not, these points definitely don't fall on the line. All right. So it really shook the biology community. And I will say that biologists remain pretty unconvinced that this is a good thing. One of the big reasons is because the conduction system that allows these impulses that depolarize the cells and cause the heart muscle to contract are really present at the very earliest stages of heart development. We've known that for a really long time. And conduction of this electrical signal through the heart tissue is critical to normal development of the heart. And so they really think, well, how is the heart limiting the contraction signal only to that area if it's dynamic suction pumping? And why? I mean, if all the architecture is there, it becomes questionable of how is it even limiting the signal to this area. And a lot of these observations in recent years have been disputed. They say that, you know, I just don't see it. I don't know how closely you looked at the movie. I've looked at that movie 100 times. I can't quite see what they want me to see, but that doesn't mean it's not there. So the other problem is that mathematicians are really struggling. So our group has been trying to drive fluid flow through an embryonic heart with dynamic suction pumping for 8 years now, and we've never been able to do it with our method. And basically this whole thing started of, why can't we make this work? We are trying really hard and we can't make this work. So what's going on here? What is missing from the story? All right, so here's where I hope you watch that really cool video that I absolutely love to send people when I talk about size and scaling in biological fluid flow, about the Reynolds number. And so we should all be familiar basically with the Reynolds number, and it's basically an inertia in fluid flow to viscosity. So this is the speed and the characteristic length and density, which gives our inertial terms, over fluid viscosity. And so if you have a bunch of Reynolds numbers compared, you can see a large whale swimming at 10 meters a second. It's got a lot of inertia. If it stops moving, if it stops forcing fluid flow, it coasts for a long time because there's lots of inertial there. Really high Reynolds number. A sperm advancing, you know, a sea urchin sperm advancing very slowly, it's got almost no inertia. Viscosity damps that out so quick that, again, if it stops moving, if it stops propelling itself, it stops applying force to that fluid, it basically stops instantly. So the big take-home picture for that is that if you, the difference here isn't just inertia versus viscosity. The difference here is if you're small, you have to, and you grow through this transition, you have to figure out how to deal with that transition and basically evolve a whole new way of getting around. It is a non-trivial transition to make across this very important Reynolds number 1, because this stuff stops working, and this stuff is the swimming and flying that you have to sort of move to at that point. So I'll just say as an aside that when you're an individual animal and you start out at 0.3 swimming and you have to get to 10,000 swimming like a squid does, becomes a really, really cool problem to try to think about how to navigate, how to prepare for that transition when it doesn't work when you are born, but you need it later. So that's a really cool aside. that you all should look up if you're interested. But what does that mean? So this is basically inertia versus viscosity. Well, what does that mean for heart tubes? It's a little bit different, right? Because the pumping of it indicates that there's some unsteadiness to the fluid flow. So we need to take into account for that, and we use a slightly different non-dimensional number called the Womersley number. And this is what the Womersley number is. And the big difference here is beat frequency. Density, fluid viscosity, and then their characteristic length instead of a steady velocity like the Reynolds number. And this is basically unsteady inertia over viscosity. So it gives the same sort of idea. Womersley number 1 is the transition. If it's over, then unsteady inertia sort of dominates. If it's lower, then viscosity dominates. It serves the same purpose. So if we look, first thing we should do is look at, and I really want to poll the audience here. And you all can just shout, who thinks that the vertebrate embryonic heart is Womersley number over 1? Can you shout at me?
Yeah, I'll go with over 1.
Okay, so how many people?
How many for over 1? 1. 1. 3. 3.
2. Okay, how many people think it's less than 1?
I'll go for less than 1, yeah.
4.
4.
Okay, so here's a chart. Here's this paper. All right, look at these numbers. Look at this. It's 0.1, and the Reynolds number is 0.01. This is a viscosity-dominated system. It really is. That you absolutely cannot ignore that. And in fact, it doesn't really start to get above 1 in terms of Womersley number until, look, this mouse is like 11, 12, 13, 14 days old. So that's off after, I believe that's even after cardiac looping. So heart tube, definitely down 1. So what happens at dynamic suction pumping at low Womersley numbers? Here is the results of a computational model in which, and I'll talk about the details of this immersed boundary method in just a few minutes, but we drove it with peristalsis, or Austin Baird and Tiffany King drove this at peristalsis and measured the fluid flow and then varied the Womersley number in different simulations. They did the same thing with dynamic suction pump. Varied the Womersley number. You can see in peristalsis it's pretty consistent. It works pretty well. Has these positive velocities here for fluid flow through the tube. Oh, look at this. This is what has been plaguing our lab for so long, right? It's a nightmare. This thing doesn't work. And this is Womersley number 1, and look at this down here. This is Womersley number 0.1, where we measured the heart tube at. It doesn't work at all. It drives virtually no flow at low Womersley numbers. So peristalsis is doing about 30 to 50 times better at Womersley number 1, and that's still way above. It's still 10 times above where these things are actually operating. So I wanted to do this dynamic suction pump video. I think it's here. Here we go. So this is this thing desperately trying to drive these fluid. What we have here is a flexible section. These little green spots are going to be where the tube pinches, where it actuates. And these are just, the white spots are just passive flow markers. So what you can see here, and the background is going to be velocity, so magnitude of velocity. And so what you can see here is that it's shaking this thing back and forth. And it's really not getting that push from inertia, right? It just goes back and forth and back and forth and back and forth. And so this represents a really, really massive problem for this system. How is dynamic suction pumping working at Womersley numbers that are consistent with those in the embryonic hearts? Just don't know, okay? So but then we have to go snap back. They saw these things that are inconsistent with peristalsis, right? They saw pulsatile blood flow. They saw peak blood velocity, and I believe this stuff, you know? They saw this nonlinear relationship. No way you can question these results. Totally unquestionable. The problem with this is that it's based on this engineer's definition, not necessarily the biologist's definition of peristalsis, all right? We got to ask, is the embryonic heart the same as a mechanical roller pump? And if it's different, what do those differences do to the flow? So first thing we have to talk about is the length of this thing, right? Roller pumps have really long lengths to diameter ratio, really high. Usually they're over 10 to 1, or 1 to 10, right? But you can see that's not quite the case here with this guy. It's pretty short. The diameter is still shorter than the length, but it's much closer to the length. And if you measure a lot of these things, what you see is that it's about 1 to 4. So it's not this huge long thing like a roller pump. It's relatively short, all right? So that's going to be important. What about small amplitude? What about the amplitude of this thing? Roller pumps usually, they don't compress the tube all the way when they operate. They don't need to, because it turns out if you do this, and occlude your tube less than 50%, you get pretty good flow that way. Don't need to. Embryonic hearts, however, you can see from these 2 inset time period stamp or from these videos that the embryonic tube really is compressing a lot, like almost all the way, much larger than 50%. Why is this a big deal? Because look, if you don't compress this all the way, you're moving fluid, sure, but you're letting a lot of fluid slide by this thing. That's going to affect how fast your mean blood flow is. It's going to affect how high those peak velocities can get, because a lot less goes through the embryonic heart when it's occluded almost completely. All right, so that's going to matter as well. So what we can say is, you know, okay, so here's what this is. It's about 90% occlusion. What happens then if we model peristalsis at the size and dimensions of this embryonic heart? What will the flow look like? What we're trying to get at is, does it really matter if we're using the engineer's definition or the biologist's definition? So is it okay to write off peristalsis based on these flow features that they've found? All right, so now this is where the immersed boundary method comes in. Immersed boundary method is a full Navier-Stokes solver. It's a direct numerical simulation. If you're interested in the write-up, it's in this Peskin 2002 paper. Takes this Navier-Stokes equation, and this isn't a big deal. I know it looks really ugly, right? It looks super ugly, but basically it's just F equals ma for a fluid. You've got density here. This is density. All these U's are velocity. X is position, and T is time. This is a pressure differential. So what this is like unsteady forces in your fluid flow. This is advection. It's a nonlinear term. It can get sort of ugly to solve. This is probably where most of the computing power goes in. This is a pressure gradient because that also drives fluid flow. This is a diffusion of momentum. Here's our dynamic viscosity here. And this is an external force. So if you push this fluid, that's where you would account for that push in these equations. And then we have this extra thing that just says the fluid is incompressible, and I know everybody's probably familiar with divergence free. So this is just saying that there's no divergence in the fluid flow, there's no mass change, there's no density change anywhere in the fluid flow, which is a pretty good assumption as long as you're under like Mach 0.8, I think, which we are. So what this method does is that it sets up our fluid on a Eulerian grid, and then we set a flexible boundary of Lagrangian points that goes through the fluid grid. Now this is great. This is a great way to do it, except when you move this flexible boundary, you have to have some way for the Eulerian grid and the Lagrangian points to talk to each other, and we do that by spreading the force onto the fluid grid with the Dirac delta function. This is what that delta function looks like. And it basically, all this is saying is that, hey, where are my closest fluid grid points? Let's apply a force, an external force here to those fluid grid points, and then solve this equation for each one of those grid points. All right. The specific implementation we're using is IBM with adaptive mesh refinement. This makes the grid really fine, close to the boundary where you care a lot about what's happening, and really coarse out here where there's not much going on. This really speeds up computation time a lot, because you can imagine this is a very expensive equation to continue to solve, and if you have to have a fine grid, then you're doing a lot of nonsense out here that you don't really care about. So that is the method. What we're going to do is non-dimensionalize a lot of these numbers, because that's what mathematicians like to do. So I'm a biologist by trade. I'm learning all these math things, and first thing they love to do is non-dimensionalize everything. So we're going to divide all our position vectors with a characteristic length, which is going to be the diameter of the tube. We're going to divide the velocity vector with a characteristic velocity, which is going to be the tube diameter times the beat frequency. And then we're going to divide time by the characteristic time period, which is 1 over the beat frequency, and we're going to use 1 as our reference beat frequency. So for initial conditions, we're going to have this racetrack circulatory system. Or is there a question?
Nope.
Oh, okay. We're going to have a racetrack circulatory system, and it's going to be a bunch of points that are very stiff and don't want to move, and then this compression system that we're going to drive with prescribed motion. This is going to have a short wavelength, just like the embryonic heart. It's going to have the tube length is going to be 4 times the tube diameter. It's going to have a large amplitude, which we're going to vary. We'll use compression ratio, which is % tube occlusion divided by 100. So 0.8 compression ratio is going to be about 80% tube occlusion. And we have a bunch of parameters we need to keep track of. We have the Wommersley number, of course. We'll take that from 0.1 to 50, so we'll span a big range across that important boundary one. We'll vary compression ratio to 40% to 95% tube occlusion. We'll vary the beat frequency a lot here from 0.25, so this spans the beat frequencies that the paper captured data over, and we'll alter the wave speed, so how fast that compression speed moves around. And I know this is a lot of things, so I have this little table that you can refer back to with all these parameters for each thing. It'll put that either up in the corner or down in the corner for each parameter sweep results we'll talk about. So the motion of the compression section we're going to drive by literally 2 sine waves. I mean, it's like the most simple peristalsis you can possibly imagine. Let me see if I can't pull this up. Here we go. So this is what it's going to look like, literally 2 sine waves, and that's it. The troughs are going to correspond so that they pinch the tube, and that's going to be it. So just as a background of here, this is the formula by which we're driving this. This is r, the distance, and it's just going to do regular sine wave. You have the frequency, the amplitude, which is going to be our contraction ratio, times d, which is the diameter, the wave speed, and the time. Like really, really straightforward, not complicated stuff. We're also going to take some measurements after the simulations are all run. Inside this racetrack, so across this little strip, we're going to measure the average speed for each simulation, the average maximum speed, so this is like very average, then we're going to say, hey, what's the maximum speed going through this thing at each time point, and let's average all those together, and then say what's the maximum of maximums, and that's going to tell us the average peak speed, all right? And then right in the center of this compression tube, we're going to look at the magnitude of the velocity, and we're going to look at the x component of the velocity, because that's going to tell us a lot about the backflow that's happening right there. And I've also have these little pictures, in case you forget which is which, where these measurements were taken. All right, so here's an example simulation, and this is what this looks like. And again, this is our racetrack. We have non-dimensional speed as red fading into white here, and white just fluid marker particles, and that's it. All right, so here we go. What's the first thing you'll notice about this? What's the velocity inside the tube doing? It's flashing. Yeah. It's pulsating, right? This is pulsatile flow. We're getting pulsatile flow and driving this thing with a sine wave, right?
Yep.
So that's right off the bat. You all can tell immediately that that is a bad assumption to make about peristaltic flow. All right, so that's thing one. And if we look at a graph of the non-dimensional speed here, for the average is going to be red and our maximum speed is going to be black. Against the simulation time, what we see really closely is this pulsatile flow. It drops all the way to 0 and then jumps all the way up. And what's the other thing y'all recognize? Oh, so this is the compression wave speed is this dotted line. What's happening? It's faster.
Yeah, it's faster.
It's faster than the wave speed, right? What on earth? What on earth? Okay? So these are really interesting results. They're really interesting results. And if we look at just the inside this compression, inside the compression tube at that dot, what we see is the UM isn't super interesting, although it does exceed that wave speed. But the UX, look at this. It's like really, really negative. So there's really significant backflow inside this compression tube. That's really quite interesting. And again, we're driving this at Wommersley number 1, which is still 10 times basically too high for the embryonic heart tube, and a compression ratio of 0.8. All right, so if we look at how that Womersley number varies, let's look at that, because we've got to explain some stuff in here. We can see really quickly that for the average velocity, for the maximum velocity, and the peak velocity are all plotted here with Womersley number for the top of the racetrack, and then we have our UM and UX here plotted within the compression tube. You can see that these peak velocities are really consistently above the compression wave speed, which is this dotted line, okay? And importantly, it's above this dotted line inside the heart tube, or here, because that's where they measured these velocities. So it's really consistent. This peristalsis is saying, hey, it's really consistent with the definition of peristalsis that biologists use. All right? And here's an interesting thing about pulsatile flow. This is maximum speed versus time for a couple different simulations. 2 are sort of low Womersley number, so this is 0.3, which is pretty close to what heart tubes operate at. This is 1, and then I have 2 that are way above 1, 25 and 50. And you can see here that for these 2 low ones, it's very pulsatile, right? And then as soon as you move up above Womersley number 10, it gets very continuous. All right, so everything is sort of continuous. It still is a little bit pulsatile, but it's at least moving the whole time. And what it's just really showing is that high Womersley number show continuous flow, like the engineering definition would predict, but low Womersley numbers absolutely do not. They absolutely really clearly show pulsatile flow. All right, so that explains one thing. The pulsatile flow is just like maybe it's just the low Womersley number part, but what about this consistent speeds above this compression wave speed? Like, what's going on with that? And I think what we need to do is take a look at that compression ratio, right? So when you look at compression ratio, so here's the same thing. We're varying compression ratio here, and then getting these non-dimensional speeds on our y-axis. We have average, max, and U peak speeds, and this is within our tube, so this is compression ratio versus UM and UX. What you see here, let me point out, low compression ratios. So if they're 0.6 and below, 60% or less compression, this pretty much follows the engineering definition, right? It keeps those peak speeds below the compression wave speed, but as soon as you get up high, as soon as you start pinching that tube really far and really hard, it starts to exceed very quickly the compression wave speed. So what this shows is that for low compression ratios, follows the engineering definition, not a problem, but all of that falls apart as soon as you start pinching the tube about around 70% occlusion, all right?
Wow.
Okay, so one more thing we have to do, and this is a little bit more complicated. We want to vary the beat frequency because this is what they did in their paper, right? They heated and cooled the embryos to vary the heartbeat frequency, and they found this nonlinear effect. So we want to do this too, but we actually want to do it 2 ways. We want to vary the wave speed, or we want to vary the frequency here, but we want to let the wave speed be proportional to the frequency. So this is like for our where on the earth did that go? I don't know. Trying to find all my videos. Oh, here we go. So if you can imagine this roller speed, the way that you would increase the frequency is just make it go really fast, right?
Right.
And so that would increase your wave, would increase the wave speed and increase the frequency. But the other way we want to do this, and oh, here we go. So this is what this looks like. This is what this looks like, all right? So you can, this is 3 times a second, so you can see it's going by pretty fast, all right? The other thing we want to do is we want to keep the wave speed constant. And how are we going to do that? The way that you do that is that you, sorry, I'm hearing my feedback, it's throwing me off a little bit, is that you change the wavelength of the wave that you're sending through, the sine wave that you're sending through. So you have the same wave speed, but just more, you increase the frequency of the peaks. Does that make sense to everyone?
Yes. Yes.
Okay, good. All right, so 2 things going on. Results. On one slide I have result. Here we go. All right, So variable wave speed. So this is where we're letting the wave speed vary proportionally to that frequency. Look at that. That's super linear. All right, linear relationship. This is predicted by that engineering definition, right? It said the relationship would be linear, and look, all three variables, hyperlinear. All right, so what about the constant wave speed? You hold that wave speed constant, and look what you see. This is remarkable, okay? You see crazy nonlinear effects. Crazy nonlinear effects by letting the beat frequency vary and keeping that wave speed the same. Look what happens when we throw up the data from that paper that I talked to you about, all right? These peaks, they attributed to resonant frequencies in that heart tube, those waves going back and forth. And what we're seeing is that we can explain that if you just decouple that wave speed from the beat frequency, you get the same peaks, all right? The same peaks and same troughs, almost spaced the same. It's kind of crazy, right? It's a little bit of a crazy result. So these non-linear relationship is really, it's not only there, but it's similar to this paper, all right? Similar to the paper. So peristalsis, large amplitude, short wavelength, just like the embryonic heart tube. How does it stack up to this engineering definition criteria? Produce continuous flow? Yeah, no. It can produce pulsatile flow as long as it's at low Womersley numbers, right? Flow speed equivalent to speed in compression wave? Nope. Peak flow speed is always exceeds wave speed for high compression rate. All you have to do is squeeze the thing more. And what about this nonlinear relationship? As long as you decouple that wave speed and peak frequency, you can get nonlinear relationships. All right, so what we've sort of said in that PeerJ paper is that the engineering definition of peristalsis is really inappropriate for evaluating peristalsis in biological pumps because all of these things happen in nature, right? Pumps at low Womersley numbers, things that squeeze really far, all right? And even this, there's a lot of evidence that says that it's really easy to decouple the frequency at which you send beats through and the wave speed. Wave speed, again, is really highly coupled to conduction velocity through tissues, which doesn't change all that much. It doesn't change in a linear relationship like roller pumps do. So what is the mechanism of the embryonic heart? All right, so we have oscillation that suggests it's dynamic suction pumping. But then we have other studies that dispute these observations. And morphology really supports active contraction through the length of this heart tube. They said their blood flow is inconsistent with peristalsis, but really if you're using the biologist's definition of peristalsis, the more inclusive definition, blood flow observations are consistent with peristalsis to a really like startling degree. And then we have this issue where we can't get it to work at these low Womersley numbers. So what is the, I don't know, y'all, it's really unclear. It's a huge debate right now. It's very contentious in the literature as well as in other places. But I do know what is really clear, and that is that this work on this problem has to adhere to relevant conditions present in this animal system. All right, that means it has to be Womersley number under 1. You can't get around that. It has to be a short tube length, has to be high tube occlusion, all right? And you got to say that it's a muscular tube with the ability, all of the hardware is there for that conduction signal to travel down its length. So if it's not traveling, you got to show that somehow, right? And you really have the potential for the critical wave speed decoupling from that heartbeat frequency. It's definitely present in tunicants. It's probably present in other things as well. So it's really, really a big unclear question right now, and I think a lot of work. I'm really excited. We're working on some other pretty exciting stuff that will hopefully chip away at some of these problems. So here's a reference list and an abstract. I can send you that. There's just a lot of stuff here, but I can send you all this if you're interested. And feel free to contact me on Twitter, through my website, on email. Stop by if you have questions and put them on our PeerJ preprint because I'd love your feedback on this. And we have some funding stuff, a lot of NSF people to thank. So that's it. I'm happy to take your questions at this point, if you have any. So let me switch back over to, not share. I can see all.
Yay!
Yay!
All right, let's give our speaker a round of applause.
Fun.
All right, so before we get going with the Q&A, I just want to announce to anyone watching online, you can either use the Google Plus Q&A feature if you're watching on Google Plus, or if you're on Twitter, use the hashtag G8O Seminar, and we'll be watching that hashtag to see for questions as well. But we'll open it up to questions here at the UCA audience. You guys have any questions?
Okay, so peristalsis and the dynamic active flow. You said that there's a lot of evidence, obviously, in your belief that it's peristalsis. But I mean, based on what you presented to us, it seems like you've actively disproved the dynamic active flow. I mean, I don't see why it's still such a controversy.
Yeah, it's very strange. Obviously, I try not to bias it too much, but I have my opinion about what it is, right? I have stared at that video a really long time, and I think a lot of other people have too, and I can't see the reflections that they're talking about, and a lot of it seems, what I see is peristalsis. What a lot of biologists see is peristalsis. There's a really big engineering community that's very wed to this idea of dynamic suction pumping. It's actually a really cool mechanism that does work really well as long as your Waldrop number is above about 10. So they're looking into doing things like active drug delivery for these things super accurate by tapping a flexible tube instead of doing a roller pump thing. It's probably much better for that. It does work really super well. You can see I made my little racetrack with my toothbrush. Of course, that's tapping really quickly, and it's much bigger than a hard tube. But we think we have a really strong case, at the very least, against dynamic suction pumping, and probably for peristalsis. I think peristalsis is much maligned in literature as being sort of an old idea whose time has passed. So I hope that answers it.
It does.
Okay, great. Any other questions?
I've got a question in terms of the model is a two-dimensional flow around the racetrack, right?
Yep.
Okay. Is in a biological system, you had mentioned earlier that there's a lot of flow resistance.
There is.
Right. In your model, it looks like it's just a racetrack. Is there a complication there, or not really?
There is a huge complication. So you're exactly right that putting resistance in the way of this thing working adds another layer of complication to it. Right now, and I can pull. I'm going to go back into screen share because I want to pull up a couple. I want to pull up a couple of videos to show you all something that we're working on. And these things, can y'all see this?
Yes.
Okay, so this right here is a juvenile tunicate that's about 7 or 8 days old. It's under a magnifying glass, a microscope, a compound microscope. Light microscopy, not a big deal. This you can see is its heart right here. And if you sort of relax your vision, you can see individual blood cells as they sort of make their way around the tube. Oh, it's reversing. So they have this cool thing where they periodically reverse the direction of their heartbeat, which is sort of awesome and a whole nother, like, really cool thing to study. But this is a close-up of that heartbeat. So here it is right here. And I don't know that anybody would argue that that's not peristalsis. It looks very much like peristalsis. So this is actually the same size and beats at approximately the same frequency as zebra heartfish do. So what we're looking at is doing a three-dimensional model of this in IBAMR right now. I'm doing a lot of microscopy work, and I'm doing a lot of so I'm working on getting these guys under a confocal microscope with tags to fluorescent tags to mark all of the aspects of their circulatory system, so we can do a 3D immersed boundary model of a resistive circulatory system that's relevant to that size and scale. I have an undergrad, Maria Williams, right now, who's doing a 2D version of this, and I can tell you we can't get dynamic section to pump work on that thing either. But we're also having a lot of trouble getting the peristalsis to work as well, and we think that it has something to do with the pericardium. So there's this outside covering that's very stiff, and that might be helping out the system as well. But she's done a tremendous job of this. This isn't. So I'll say that this is more of a C++ library than it is like a program that you just go and draw lines in. So she's having to draw the geometry out in MATLAB. She's having to make sure that it has the right number of points. She's having to write the C code to make the wave propagate. And it's a tremendous amount of work, and so she's still working on it. But we're very close to having something work, which is pretty exciting. So we're hoping to get that resistant circulatory system and stick peristalsis on there, and also dynamic suction pumping, and operate it somewhere close to Weimer's line number 0.1 or 0.3 or something like that. That's in the range. So it is a big problem. It's one that we're sort of incrementally getting to, but I have my very strong doubts that dynamic suction pumping will work any better when it comes up against a huge amount of resistance.
Right. It's remarkable. I mean, that's just an incredibly, your presentation is just amazing. It's just beautiful to see that mathematically and then computationally working out with such beauty for the data that was taken. That's just gorgeous.
It completely shocked me as well. I'm not into things looking that pretty because I do experiments on animals, which A, never cooperate, and B, never look that. So if you want to do bio stuff, just I know it's just a complete mess all the time, and nothing works like it should. But when it does, it works, and it's awesome.
Cool.
So, are there?
You guys have any more questions here? We'll check over on the Google Hangout and on Twitter as well. Let's see if we have any questions over there. You guys have any questions here in the room?
I do.
All right, fire away, Katie.
I'm actually a biology major, and whenever you were showing the segments on peristalsis, this may be silly, but I couldn't help notice the similarities between the movement through the sections and that of the movement of worms and how worms move. So I'm on the right track with this.
Yeah, no, their movement has been described as peristalsis as well. So it's the same kind of like compression, except what they're doing is they're moving fluid, but they're moving a hydrostatic skeleton. So their worms are basically a bag of goo, watery, surrounded by muscles. And they do this in order to move, so it's a little lopsided. It's not quite a perfect sine wave, because if they did, they wouldn't be able to locomote. But it's a little bit of a lopsided move, but it's definitely very, very similar to that. So it's a very widespread sort of movement in biology, and has caught a lot of I don't know, track lately for being sort of an old-timers concept. But I think what I really wanted to name this paper that we just put out was Peristalsis. It's more than you think. But I got nixed on that, so. Gotta sound more serious, she said.
Okay. Cool. Any other questions from the peanut gallery here? Well, we always can ask questions, right? You have one you want to ask?
Yeah, one of the questions that we usually like to ask is, you're obviously, you're into biology, and you're doing biology, just now started and you're getting into math and stuff. What was your life and road to this career path and getting into sciences?
Oh, that's a good question. It hasn't been linear for sure. I entered college really very certain that I was going to become a veterinarian. And, that fell apart, I think, my first semester when I did not make a good set of grades. I had a really pretty rough transition. I came from a pretty rural high school in North Carolina, and so I wasn't 100% prepared to come into college, and so that made me rethink things pretty quickly. I took a physics class Actually, it does have stuff to do with waves. It was a music of physics, like freshman seminar, and I really loved it. And I love the idea of what really fascinated me was psychoacoustics. If you know church bells, a bell tuned to an A doesn't actually produce any of the wavelengths that are associated with the piano producing the same chord. So I found that very, very fascinating. Like, how does your brain construct this thing that it's not actually hearing? So I ended up doing some undergrad research in psychoacoustics, decided I could not work with human subjects. They were too difficult, and you can't just stab them with things like you can get in trouble. I don't know but I took a class as a junior by somebody who I admire very much, William Keir, here in the biology department, and he had a comparative biomechanics, and so I was always on the fence of like, oh, I really love physics, and oh, I really love biology, and oh, but they're so separate. And this all of a sudden just snapped it together, you know. To snap those things together, and it was all about how things run and how they fly and how blood gets pumped and how noses sniff and all this stuff. And it was so fantastic that I decided I needed to go to grad school in that. And so I applied to Berkeley and got in and ended up doing weird math thing. And now nobody knows what I do, and I can't describe myself. And I have no idea how that will get me a job later. I am having fun with the things that I do right now, and I think that's the important thing.
Are you considering like academic type jobs, or? I
am. I think I'm sort of going on the job market. We'll see how that works. For biology, it's different than I think physics and math right now. It's very competitive, and I'm still very junior for the job, even for the job market after being a postdoc for 3 years. So we'll see how that goes. I'm about to move to UC Immersed for a second postdoc in another math department, and we're going to work on a lot of math modeling stuff and experimental stuff as well, which will be exciting. But I'm really interested in teaching. We just had this big symposium at our biomechanics type meeting, in which we were trying to get people convinced and excited about active learning strategies. So I'm also very interested in teaching. So I'm thinking maybe smaller research, much smaller research university, not like a UNC or a Berkeley type size, but sort of a smaller one. But it could allow me to manage more undergrad research projects, which I absolutely love. I love working with undergrad researchers because they have so many great ideas and they're so excited about doing stuff. So that's kind of where I'm aiming. Will I end up there? Not sure, but like I said, I'm having a lot of fun with what I'm doing now. So
cool. It definitely comes through.
Great. Well, I will have to say, so some people get very quiet when they get nervous, and I get very enthusiastic with my head. This is partially just being nervous.
Well, what a lot of smaller departments in physics are doing are looking at intersections between different fields to try to help maintain the student body. And so, like Katie mentioned, we've got a biophysics program here, and you'll find that that's becoming more and more common in physics departments. And so definitely look at, a biophysics type program, because you would fit right in with your research that you're doing, your background in fluid mechanics, fluid dynamics, and computational fluid modeling. You could walk the walk, no problem.
Yeah.
That's perfect. And we're really, often these departments are looking for people that can bridge those disciplines, and you're right in that sweet spot.
I do not know where I fit at this point. Often people ask me what I do, and I'm like, I really don't know. I mostly sit in front of a computer.
Yeah. Well, I think that's the future. These departments that are very siloed. I think that we'll see that, in the 10, 50 years that go forward, that will have to bleed away a little bit. That these combinations of fields are really where the real work needs to be done. And so positioned yourself in a good place to start with that.
Great.
What, so in terms of your programming, when we did our initial test run, I kind of asked you this question, but for the students, what do you use to write the computational fluid code in, and then is that running on like a desktop machine, or is that going to a supercomputer? How are these computations being done?
So the computation is a big, huge set of it's a big, huge fluid solver that I'm using that somebody else has written, and it's an implementation of this immersed boundary method, which Vertebrate has made this adaptive mesh refinement that super speeds up computation time. So, but still, it speeds up the computation time, but I can't run it on my desktop computer because it would take literally months for it to run. This is a parallelized version of the software too, so we can work on several processors. I write most of my geometry code in MATLAB, so I've been coding in MATLAB for, I don't know how long, like 5 years or 6 years or something like that. That ends up being pretty good to generate the files that we need for telling the target points where to be and the vertex points where to be, all those Lagrangian points. But the thing runs on our cluster here at UNC, which is a really big, powerful cluster. I don't even know how many processors it has. It has lots of processors. But usually when I run a single simulation, I'll use up to 16 or 32 processors, and it'll run for 24 or 48 hours. And that's a 2D simulation. So once you get into 3D, you start, you know, it multiplies quite quickly. The amount of time that you need to use. So what I do is I upload things on the server. We call it Kill Devil because you can see. And so I'll upload my files onto Kill Devil, and there's an IBAMer sort of, I can't remember what they call them, sort of unit that you can load up, and it will run it batch processing on these things. And so I know a little bit of C because to write sort of how you move the target points is all written in C usually. So I know a little bit of C, and I've been fiddling around with that. And then I do all my data processing in R. I've left MATLAB behind because it's so aggravating. So half of the time, R gives you really pretty plots. So yeah, and I also use a program called Visit, which all of those movies that you saw were made in Visit, and it's an extremely powerful 2 and 3D fluid visualization program, data analysis program. So lots of different things that I've had to figure out how to use. That's been most of the first year of my postdoc, was just coming up to speed with a lot of these things. Even though I had some computational background, I had done Markov chain Monte Carlos and stuff in MATLAB, and those were big, huge simulations. It's different to move it to something like this. So it's pretty common.
Well, what advice might you have for current undergraduates? What sort of things do you tell your undergraduate interns that are working with you, and what advice might you have for any undergrads that are watching or here in the audience?
So it 100% depends on what you do, and it doesn't at the same time, and I'll kind of explain that comment. If you want to do research, you should be doing research. It's like what they say about writing. If you want to be a writer, write. If you want to do research, do research. You know, find something that interests you and get into a lab somehow to do it. You know, I know that from my experience in grad school is what a lot of people like to see. You know, and this varies a lot, I think. I don't want to get hammered on Twitter because people have different opinions, but it matters that you do research and you kind of understand what it's about, and you understand that it's mostly being frustrated all the time. But just having that experience speaks volumes, I think, if you're interested in applying to grad school. I would say that don't shoot low if you're interested in applying to grad school, because I did not think that I had the grades, the GREs, or the experience to get into Berkeley, and I did it anyway and got in. So if you send your application to Brown or Cornell or Berkeley or wherever, the worst they can do is reject it.
Right.
You might even get some feedback about how to make it better for your next round. So don't feel like you can't go to those big schools if you're not at a big school already. Just go for it. And I think that if you're not interested in research, like you're interested in going to med school, I have a lot of people that I work with who ultimately go to med school, and it's great. Because a lot of the people I work with are wonderful people. They're extremely compassionate and they're extremely intelligent. And I would say I would want that type of person to be my doctor. So, it's great to do these type of research projects too, I think, if you can get into a lab that will allow you to develop your own sort of ideas with it, because it is that ability to stick things out, to solve big problems that aren't immediate. You know, the answer isn't in the back of the textbook sort of thing. And just having the ability to tolerate being frustrated, I think, will take you very far in life. and research, for the most part, is partially about being frustrated, especially if you do research, say, with computers. I take an exercise class, and once a week I get to smash a medicine ball into the floor, and I just imagine it's my laptop. So, yeah, so get involved in undergrad research, especially if you want to do research. And some people don't like it. Some people I've worked with have just said, it's too frustrating to me, I don't like it, I like to work with people, and I feel like I'm crammed inside a dark room on a computer too much. And that's great to know, too. It's great to know that that's not what you want to do. And it's fine. Like, I know that I don't like analytical chemistry at all, but it's great that other people do. It's great that that's what you want to do. I know that a lot of people don't want to, clean out tanks and animal snot and all the things that I deal with too. So, and I go over. So yeah. Any other questions about stuff? Anything more specific about bridging two fields, or if you're interested in getting into a specific program or anything like that?
Well, do you have, or do you know of any REUs there at UNC, either in your group or in sort of the department there?
Yeah, so the physics department has an REU, and I don't know the stat. I don't know when those applications are due. There is also a really great, if you're interested in mathematical biology, which is not necessarily, fluid like what I do, but a lot of sort of network stuff or disease modeling and things like this with cool math in it. There is a program at UNC Greensboro that's run by a friend of mine who's really excellent, and I've heard really great things about that program as well. And all of these things are supported. You live there. Greensboro is I guess Chapel Hill's not really a super exciting town either, but the programs are really nice. So UNC Greensboro has a really nice one, and those are the two that I know about in my area. But there are a ton all around the country doing just about anything you can imagine that you want to do. So kind of whatever interests you, look on the RU website and try to find something. And I would say do it pretty soon, because those deadlines are starting to pass. So is anybody out there, are you already doing an RU, or are you thinking about doing an RU soon?
I applied to several, but I haven't heard back. So.
Okay.
I know that a friend of mine, Tony, he got into an RU there in New York for something about education. So.
Cool.
Yeah, we really encourage our undergrads to apply and start applying early, like in your the summer of your sophomore year, so that then you can maybe go to a local or close one first, and then use that to help bootstrap your way into one farther away, maybe the next year. And so if you're lucky, you might get to go to two before you graduate. That really helps get experience at, oh, this is what a different lab is like. This is a different sort of
because the experience that you will have will vary so incredibly largely that it's almost difficult to paint a broad picture of it. So my lab was dramatically different than labs at Berkeley that were within our same subgroup. You know, just not only the culture, but how you got funded, how much teaching you had to do, the types of projects that you worked on. We had an incredible amount of independence, an almost distressing amount of independence in my lab, to pick your own project and also to fund your own project, whereas another lab that was close by, you worked on the projects that the PI wanted you to work on, but you were really well funded and had all sorts of access to cool equipment. So it's a really, I mean, it's difficult to kind of sum it up because there's so many different experiences that you can have.
Yeah. Do you feel more at home in a mathematical department or in a biology department, or are you alien in both?
I think I am truly alien in both at this point. I would have said a biology department, but what I do is often viewed by traditional biologists as not biology. And I most certainly do not do new mathematics. I do implementations that have already been well formed and tested, and I'm using them to answer fundamentally biological questions. So I can work really, I think my strong point is that I can work really well with biologists who are very much more into the biology than I am, and I can work with very, very applied mathematicians who have maybe a tangential interest in biology work. So I attempt to actively bridge those two fields as much as I can. I see myself almost more as a facilitator of ideas going across the boundary than doing work on either side. But this is sort of it's hard to convince people to give you money to do that. The funding structure is not quite caught up to where I sort of delicately balance on the two sides. So
interesting. It sounds similar. I have a colleague and a friend who is in computational chemistry, and they're sort of also alien in their own departments because chemists tend to not trust what the computer says, and well, it's quantum mechanics, and so you can freely talk to physicists. You know, it's like, okay, sure, I understand what you're doing. Computational chemistry is basically physics, but it can be alienating.
Yeah,
so,
it's a little weird. I was very much the weird kid in high school who didn't have a click, so I'm kind of used to it. So.
See, all the more reason to come to physics because we're the same way. We're all the weird kids.
So I'm kind of used to floating in between clicks. That was very high school for me. So that's fine.
Well, if there are no more questions, we're good? All right, let's thank our speaker one more time.
I had such a fun time hanging out with you all. I really do get inordinately excited about peristalsis and tiny things. So thanks for letting me sort of vent my excitement towards you.
Well, this is fantastic. So we really appreciate your time, and this has been a great seminar. Thank you very much, and I'll be sending you a follow-up email today or tomorrow.
Thanks so much, and good luck to you all, whatever you end up doing.
Thank you very much. You too. Bye-bye.
Bye.